recording. So this was born out of the Society for Study of Evolution's really nice initiative to start, research working groups. And Matheus and I decided to submit one about budding speciation. We put together what we believe is a very exciting group of people to talk about budding speciation and a lot of interesting, viewpoints. So thank you so much for coming. I think you're going to really enjoy the talks. Make sure if you want to look at captions to click the more there and click at show captions. And if you have any questions at any time, you can write something down on the Q&A section. And once we get to the questions portions with our speakers, we'll get to it. And please make sure to stay around for our panel at the end, our last 30 minutes. And with that, I'd like to introduce our first speaker, Matheus, my co-organizer. He's going to be telling us about a lot of his exciting work with islands and extirpation and all we can learn about budding speciation. And he's also going to do a nice introduction to our symposium. So take it away, Matheus.
Can you guys see my screen okay? Okay. Nice. Thank you, Bruno. Thank you, everyone that show up to our symposium here. Is really exciting to be the first person presenting here. I just want to quickly dwell on the menu that we have in both our first session now and our second session tomorrow morning, if you are in standard time. Thanks again for the society for giving us a space and for encouraging our work. And long story short, budding speciation is this speciation mode where the mother lineage does not dissolve into two lineages. So we have like a very clear daughter lineage and a mother lineage that gave rise to that. So, and why usually we do not even think much about budding speciation, right? I think this was like very succinctly put into one of recent papers by Tomo, which is presenting the next session, saying that like the clades were adamant that like these sorts of relationships, daughter-mother lineage relationships, could not be scientifically tested. So they decided to use just the ambiguous cladograms, just bifurcating graphs. And due to this, like some sort of like way of seeing things, we usually don't think much about budding speciation. And other speakers like Danielle that will say later in this session here will say like many times actually we don't need to bother too much about this because our comparative methods, for instance, they assume Markovian process. They have no sort of memory of what it was the mother lineage before. And that's a fair way to think of it. But if there is some sort of asymmetric speciation or asymmetric trait evolution when it comes to which lineage inherits which trait. This is a phylogeny drawn in one of Danielle's paper, and the yellow trait is the trait of the mother lineage, but it gives origin to the black lineage here. And if we use like simple likelihood reconstructions that assume bifurcations, we will reconstruct the history of this trait evolution quite wrongly, actually. So this is one potential problem of ignoring budding, and there are others too. In fossil data sets, we have like temporal durations of species, and if you have to accept a fully bifurcating phylogeny, many times we have to stretch these durations back into the past, creating what we call ghost lineages. And this will change our impression of divergence times and the rates of bifurcations, for instance. So accepting budding allows us to relax this quite strong assumption or bias that we have to sometimes have. There are other consequences which other speakers will explore here. Professor Brandvain and Professor Kunert will explore what we gain when we try to explore directly these ancestor descendant relationships. And the other speakers we also will explore what can we learn about trees, how we can improve phylogenetic inference methods with thinking, if we allow ourselves to think about budding, and how we test evolutionary hypotheses related to this idea. And some of these will be in the next session, though. So this finally brings me to my actual presentation here, which I was arguing with you guys. The landscape fragmentation reveals that there is some phylogenetically widespread budding speciation potential in squamate reptiles. I'm now a postdoc in the Institute for Global Change Biology in the University of Michigan, so thank for them for paying my salary for a little while now. And I want to start the talk talking about this gecko here, Cyrtodactylus tiomonensis. It's a tiny gecko that lives in a tiny island in the region of Sundaland in Malaysia. So this is the region, in case you don't know. And if we zoom in close to the Malaysian territory here, you see this very tiny island called Tioman, and that's the only place in the earth that you can find that gecko. But actually, Tioman was not always an island. So this is a GIF showing our planet since the last glacial maximum. Wow, the GIF. Well, okay, we have to wait until the GIF. I don't know why the GIF. Okay, now it's from the beginning. Okay, so this is a GIF showing our planet since the last glacial maximum. The colors are just temperature. I want you to pay attention to the black lines. So as the world glaciers melted, I will come back, and you see in the next loop of the GIF here. As the glaciers melted, the sea level rose, and what was previously a continuous mainland became an island. We call these features land bridge islands because they were connected with the mainland, and they stranded many organisms in these regions here. But this is a very recent event, just 25,000 years ago. And this was a decent change in sea level, more than around 140 meters, depending on how you estimate. But the point here is that Tioman was not an island 11,000 years ago. It was just another coastal zone of the mainland in Malaysia. But around 9,000 years ago, it was already an island. And actually, this gecko is not the only one in the genus that is endemic to a land bridge island. Actually, there's another lineage closely related to this, Cyrtodactyl celibuatensis, which occurs in the Celibuta Island here, which is also land bridge. So, but this doesn't end up in geckos. So there's like these two insular endemic vipers in Brazil that also occur in land bridge islands. And when people do phylogeographic studies, usually these lineages show up nested within the continental bothrops, which is very widespread throughout the coastal region of Brazil, in their region of Brazil. And so my point here, I was wondering, and I want to discuss with this talker, how broad phylogenetic this type of pattern is. Like, I just want to have like some sort of like a bigger picture, some sort of like exploratory or phylogenetic natural history analysis of these types of phenomena. So I want to know how these endemics are distributed, and I want to see if there's any clear pattern between speciation rates or trait evolution in these special lineages over here. So first, where are they? I cleaned some data here, and this is basically the diversity longitudinal trends in these organisms. Is not most of it is in Sundaland and in Sahul regions, but you have a few lineages in the Americas and very few ones in Africa. Still, though, we have these endemics in many of the realms of the vertebrate realms of the planet. When it comes to phylogeny, we also want to ask, okay, where are they phylogenetic? Do some clades have some greater tendency to produce these lineages? And with some comparative methods, we can test this. And basically, they are very widespread, phylogenetically speaking, because you already saw the geographic pattern, so you can tell why there are so many red and so many pink species. They are just Indo-Malay and Australasian lineages. But when it comes to like figure like what is the rate in which this trait appears, is actually quite fast and appears many times in the squamate tree. How does this species look like? Well, 42% of them are sister species to a mainland species, 20% of them are sister to another land bridge island. And by the way, I removed all the lineages which the whole genus would be in a land bridge island. So this is still like under the story that I'm trying to tell here. And 37%, like a third or so, I put that in the others basket, which they have different types of like topology in which they appear in the topology, so to speak. So I want to have an even better like idea of what's going on here. So I tested if we can predict these lineages with a binary Bernoulli PGLS using speciation rate and body size as predictors here. So long story short, technically speaking, both speciation and size are significant. but the effect size associated with the speciation rate is actually quite small. So if you see here, that's the color. So usually the green and the orange bars are very close to each other, but the big jumps are like really if you were a lizard, lizards have like about half of the capacity to produce these endemic lineages compared to snakes. The big leaps there. Actually, snakes do a lot of trade evolution compared to other types of lizards. So this is to some extent aligned with previous finds about trade evolution in squamates, but still body size is a decent predictor too of these lineages. If you half your size, which is the circle symbols here on the left, you also increase your chance to produce yours Landbridge Island endemic. So basically, these organisms usually come from smaller lineages. But another thing that I want to say is like, okay, they come from smaller lineages, but are themselves like dwarfs or smaller species in general. And it happens that they are. Like, this is just a quick effect size. So basically, I'm comparing this using the variation in the species in that whole geographic radiation. If I pick the monophyletic clade in that realm, um, how do the endemic look like? And usually they look like smaller versions of the radiations. All these bars to the left in this graph here. There are exceptions, but most of them are very small compared to the radiations in which they are located in the phylogeny here. So this looks a little compelling to me to say that maybe some of these lineages may be nested in the mainland lineages. They may be good candidates for budding speciation. We have to look closely, do detailed phylogeographic analysis. But I think it's interesting to look at these lineages because they may have a directed inheritance of trait evolution. They are smaller lineages and with size may come a lot of other different traits associated with that. So for instance, the Botrops example that I presented in the beginning, they are smaller versions of the continental snake, for instance. Uh, and before I finish, I just want to say that this is not perfect. Of course, like this, if you count the terminal, if you measure the length of the terminal branch length, most of these, virtually all of them, are much longer. So the split was estimated to have happened before the last glacial maximum. So maybe not all they were formed by the last glacial maximum, but the sea level fluctuated for a long time in around that sea level. So maybe these species remain some sort of cohesive, and they still raise questions about the cohesion of these species. So anyways, I think they're good candidates for you to study this phenomenon in living organisms. Usually, budding speciation is this phenomenon that is mostly attacked with fossil data, but maybe we can also look at some living lineages too. This is kind of my take-home message here. So before I finish, they are about 2% of squamate biodiversity. We can investigate them like in more detailed analysis than what I did in this talk here. Maybe we can learn a little more about budding speciation from these awesome lineages. And what we know for now is that most of them in Sudan or in Sahul, or the lands associated with these two supercontinents, their phylogenetic distribution is random. So it's much more dispersed in the phylogeny than any Brownian motion or any trade evolution model. They are more common in snakes, and they usually lead and come from smaller lineages. That's what I have. Thanks for everyone that helped me with the data and with the idea. This is how you can find me. And thanks everyone that fund my activities here at Michigan. And thanks for SSE again for giving us this structure to sit down and discuss these ideas on budding speciation. Thank you.
Nice. Thank you so much, Matheus, for your talk. We have maybe a time for one or two questions. If anyone has anything they wanted to ask Matheus, feel free to drop it on the Q&A chat, or if you want to use the raise your hand react, I can unmute you.
We'll also have the panel discussion at the end. Go, Laura.
Matheus, great talk. I
Thank
was
you.
just curious, have you thought about how taxonomy might influence like these results? Like, do you think like the oversplitting problem or practice might potentially be affecting more snakes than lizards? I have no idea, just wondering. What are your thoughts?
Yeah, right. Absolutely. I think a lot about like how, what is the possibility of these lineages just coalesce because we have a monophyletic perspective of how species should look like. We Some people will call them species, and this creates the impression that these lineages are special. And I don't have a final answer. Most of these lineages are studied with like mitochondrial DNA, for all I can tell. Like reading the papers, reading the phylogeography papers, they're usually using like a few markers. So I don't have any like sort of a generality to offer about like what is the like what is how substantial is the evidence that this species should be called species. This is clearly a big limitation. And when you look at the terminal branch length that I showed, this becomes even more um I don't know, between suspicious and interesting. And so I think it's something that we have to think about, but I don't have like a very clear answer. What should we do? I'm very interested to hear what Yaniv has to say about this phenomenon. Like, what would be the sort of evidence that would allow us to say, no, like this is like definitely species that budded off from the mainland
Which is a great
lineage.
which is a great segue because we now have our next talk. Thank you so much, Matheus, and thank you, Laura, for your question. Now, Dr. Yaniv Brandvain is going to tell us a little more about this budding detection in more recent divergences. So, take it away, Dr. Brandvain. Oh, you're muted.
Thanks. That was a really fun talk. And yeah, unfortunately, as some talks go, mine was more aspirational. So I don't know how far we'll get with this. I know we won't get far with this detecting today, but it gave me a time to reflect, presenting this talk on what we mean when we say budding speciation, what we're trying to see, and what the process is. And that question about taxonomy, I think, is quite relevant. So it was a fun lead up. Um, because yeah, well, so I started to think about budding, and then I thought, okay, what's a bifurcating tree? And I don't quite know like how we have bifurcation. I would say it's something like this, maybe. And please, I hope I'll have time for questions or telling me I'm wrong or something. But it's something like that, right? So you have a population, you somehow magically split in half by a vicariance event, or maybe that's on the edge or something. And then you diverge in allopatry. You get different. That's what those two colors are. One of two things happens. You diverge and you're reproductively isolated, so you're two different things that can now hang out in your two different species, or you'd have not enough divergence, populations merge, you go back into one, and then you wait for a new vicariance event to have another shot at this speciation process. And you keep doing that over and over and over again until diversity and extinction and so on shapes your bifurcating tree. So please correct me, or I'd love to hear thoughts after with my remaining time about if that's what we mean when we think about a bifurcating model. Darwin's model, of course, was a little bit more complicated. He's got like polytomies and stuff, and he saw a role for extinction. I mean, this does not look like any um, bifurcating tree that I've seen in a scientific publication, except for people talking about Darwin. But like, you know, it still seems like somewhat cleanish splits. I think he didn't really have, you know, uh, I mean, he knew about hybridization, but he didn't quite know about like gene flow or integration. He didn't know what a gene was really. So I think it was harder to conceptualize the modern ideas of population structure and what budding speciation might be when you don't quite understand much of that. Okay, so again, this is just my sort of view on thinking about what budding speciation might be. So I think we start, instead of thinking about starting with a panmictic population, you might think about lots of populations. Here's a stepping stone model, but some metapopulation structure or some substructure within your large mothership population. A population becomes isolated, diverges, and then, a few things could happen. if all populations here kind of coalesce, just divergence here, divergence there, divergence here in the orange lineage, and these all coalesce and kind of homogenize, right? That was a not homogenized, but kind of get equal, that signal is lost of that close relationship ancestrally. You had a budding event, but I think there wouldn't be any trace of that in the lineage, in the history, in the phylogeny. I don't think you find any sequence evidence of that, although maybe I'm wrong. Um, if the orange goes extinct, of course, it looks like nothing happened. Um, but if a new lineage in green becomes isolated while other bits persist, I think then we'd have a long-term signal of budding in our phylogeny, and we'd be able to look at this. So that's just my hunch, and of course, it need not be one species. You could have a cut that would take three, one population. You have a cut that could take two of these, or one could jump somewhere, or so on and so forth. So that's my rough view of what I think budding speciation is, and happy to talk about that more as we go. Okay, so oh no, I wish I had time. Um, so how are we going to look at this from budding from the species from the view of population genetics? So I'm going to just look at this one event. In some cases, we might happen to see more. I've worked on the Mimulus guttatus species complex, where it seems like there's just lots of buds all over the place. But I think we're learning about this process one group at a time. So we're looking at this orange thing, and we want to look at that to understand the process of budding. Okay, and so I think to find a signature from population genetic data, you'd want to find a bud that's not equally diverged to all mothership populations. So you find um, a species or two species, and you'd say, okay, within, this one on the range and the edge here, it's more closely related to something near it than the rest of that mothership population. Um, and you'd want that to be closer than pi across that mothership, and you'd want to be certain that is generated by the historical process of speciation and not by secondary contact that will bring that group closer. So I think that would be sort of reasonable evidence for a budding event in an extant group that we're looking at. And so I'm gonna talk about this Mimulus paper where I worked on a couple years ago that had me thinking about this, and that is the model I have in my head as I hope to go and do some more formal inference. So we have a selfing species, Mimulus nasutus. It's outcrossing relative guttatus. We think that nasutus is a bud off of the guttatus lineage. You'll see that in a minute. Here's their range. Guttatus has a broad range. Nasutus is within that range. There's regions of micro sympatry. So these two DPR populations and CAC populations have both been studied extensively by my collaborator, Andrea Swigart, at University of Georgia. Okay, so we can look at these data as a few different ways. You can measure pairwise sequence divergence. Mimulus are remarkably diverged, and you can see that Nasutus has relatively little diversity, and that it's more closely related to Guttatus than Guttatus is to itself on the whole. You can see here that Nasutus is nested within this bluish lineage corresponds to the kind of south western, kind of separated by the Sierras, the southwestern bit of the Guttatus group, and is more distant from the northern group. And so I would think about Nasutus as budding from within the southern area of Guttatus. So you can see that this event was associated with an extreme reduction in diversity, as we see um, in the origin of sulfurs. And then there's some cases of deeper coalescence. So this is a histogram of diversity within the sulfur on a log scale, so lots of the diversity is really, really small. Some of it is deep and deep coalescence. Okay, so you can look within Gutatus, that's quite deeper coalescence, broad diversity. And in Allopatry, you can see between species and Allopatry, it's also quite deep. You can see here's in Sympatry. And this is the northern and southern one. And so what you can see is you can see two distinct processes that are going on here. So the first bit you can see is that the southern sample has a peak of divergence here that is not particularly diverged, well, or less diverged than within the northern one. And so we think that's our evidence of budding. And there's a few different, this is the best way I showed it in this plot here, but in many ways we can see that that Nasutus is more closely related to the southern Gutatus here. You can also see that there's also been gene flow here. You can see that from that uptick here of very recent coalescence between the species in sympatry. And so we can split out that process of gene flow from ancestral population structure. This is the ancestral population structure. These two peaks here. This is the gene flow. Okay. Um, okay. Yeah. So there we go. So the takeaway here is that we're seeing budding speciation and gene flow simultaneously. And more generally, this seems to be the mode of speciation, the Mimulus Gutatus complex. There's, depending on the question about taxonomy, there's an unknown number of species depending on who counts. And they all seem to be basically a Gutatus-like thing with some ecological adaptation or something that gave somebody the idea to name them something else. But these are also, reproductively isolated often. So they do have intrinsic barriers between them. So this seems to be the model of speciation in the Mimulus Gutatus complex and might be a general model of speciation in many complexes, whatever that means. I've also looked in other taxa. So in two other selfing outcrossing systems, we roughly see the same sort of story, although not always as clean, but we see a budding model followed by secondary contact. I collaborated with Suzanne McGaugh on Astyanax Mexican, the cave fish, and we see basically the same pattern there. We see a small any bud with gene flow. Okay, and so I was hoping to give this talk about this method development, but sadly, we didn't get far enough. But given potential budding and secondary gene flow, we want to learn was there budding? Is there ongoing gene flow? So we want to be able to see if both those things are happening. If we can find this, we want to know the time and place of initial budding, and we want to know the time, place, and extent and direction of secondary gene flow. And I'm not going to bore you with things. We wrote down an equation. It's long, it won't inspire you. We can calculate likelihoods. We can do some inference. We can simulate under the model. We can often recover parameters, but not always. And we're at that stage of sort of method development. So I'm hoping to keep you posted about future progress. But I'm excited to try it out on these and some other systems if anyone has data they would like to discuss. And with that, I have a few outstanding questions. Um, uh, so how common is budding speciation? Is it the exception or the rule? And part of me wonders, like, if my first view of what bifurcating a bifurcating model is, which is just a really clean split, um, is what we think it is, then like, is there ever not budding speciation? And it's just like a question about, if that signal is maintained long enough or it's covered up by secondary, by gene exchange within the major population. Another question is like I've been studying these macroevolutionary buds, these selfing lineages, these cave fish, these things that look kind of wimpy and perhaps destined to extinction. So is that the way that we see these, stronger evidences of budding? And if so, does that have any real evolutionary impacts on long-term diversity, or is it just things that blip in and out? And then, yeah, when and how is the signature preserved on phylogenetic timescales? And what if I think can erase that signature? And I've been thinking about that as I put those little circles together. So those are my big questions and ideas. And with that, I would like to take any questions from you or have any more discussion. And thanks for bearing with me as I have my my preliminary stuff together.
Thank you so much, Dr. Brandvain. I think, unfortunately, we don't have a lot of time for questions, but if you have questions about Dr. Brandvain's talk, please stick around for our little panel at the end. And with that, we're going to go to our next speaker, Dr. Daniel Caetano, who's going to tell us all about the consequences of this pesky budding speciation on comparative methods. So take it away, Daniel.
All right. Okay, how is it looking there? I cannot see you, but it's okay. All right, so hopefully it's looking good. Otherwise, stop me. Good. Great, thank you. All right, so first I would like to thank Matheus and Bruno to invite me for this conversation. I think it's a very exciting topics. And my talk is going to be from the point of view of comparative methods. So how budding can affect what we do and which sort of questions and models we need to think about it, right? Such as Matheus started the conversation, right? We can think about budding as the recognition of the progenitor lineage after speciation, like I'm showing here on the top, right? So if we have lineage A as the ancestor, and then we can recognize lineage A after the event of speciation, then we might be seeing budding. Bifurcation then is the case in which we cannot do that, right? We cannot recognize lineage A after the speciation event, and we end up with these two different lineages, C and B. But one thing that I think is important thinking about comparative methods is what recognizing the continuation of a lineage means for the lineage. And in this talk, I'm going to talk a little bit about age, but coming next. How do we identify these budding events, right? So here we can think about fossil records. We have lineage A, B, and C. We have the information from the phylogeny. So we know that C and B forms a group, and then A together with B and C also form a group. What we have of evidence is overlapping time, such that we know the B here, the progenitor, occurred first, and it still can be recognized after these two budding events, right? And I'm here going to talk about this lineage B, right, as the progenitor lineage, and the other ones as the daughter lineages. Now, what is interesting about budding speciation, in my opinion, is that it's a term that's often used in the paleontological literature, but not that much in the neontological one with molecular trees. So can we even find budding speciation with molecular trees? And I think that we do. So here we have an example, right, in which we have 4 species and they are represented with different samples from different populations. So we don't have only one terminal for each one of these 4 species. And what I'm showing here is a case in which we have species 2 closely related with a subset of species 1, making species 1 paraphyletic. And this might be evidence for budding right there. But what might be happening is that many of the trees that we see in neontological work do not have such a detailed sampling that we would see in phylogeography work. And we also have some questions of taxonomy, right? Maybe the pattern that I'm showing here is something that gets filtered out during the work of someone, end up not showing on the publications because nomenclature itself can change this, right? Can make species 1 monophyletic. Now, thinking about that, we made many simulations and we tested many models. So I want to point you to this paper that we published in SysBio with more information. But here I'm going to talk about most of the highlights, right? One thing that I like to think is diversification models and how budding can change that, right? So one thing that I think is clear and kind of intuitive is that if we think about the tree as a bifurcating process or a budding process, it will really not affect the number of species that will accumulate over time. But like Matheus introduced it too, if we decide, for example, here on the top, to resolve these speciation events as budding, we'll introduce less of these ghost lineage effects, right? Which is this extension of the fossil occurrence of the lineage back in time, such that the most recent common ancestor of all the lineages are at least as old as the oldest one. I made here on the bottom this lineage through time plot, and what you see in this example is that thinking about bifurcating will push some of these nodes to be older. This can affect then the age of the nodes and might affect our burst of speciation and extinction. So if we are thinking about models of diversification and we are asking questions about, for example, how traits affect diversification, a budding tree and a bifurcating tree can potentially change that, right? Depending on how often budding happen and even if we have evidence for that in our trees. There's a really cool paper that I recommend. It's this one by Nicholas Crouch and Proc B. And what they did is that they did bifurcating and budding on bivalve families, and they really show how that affects the node ages. It's a really cool paper. Now, switching to trait evolution models, which I think is also really cool to ask this question, is interesting the fact that trait evolution were based in this idea that each branch of the tree is evolving independently, right? And this is actually a really important thing for the math of these models. So if you think about independent contrast, if you think about Brownian motions, be it a homogeneous Brownian motion or a Brownian motion with changes in rates over time or not, just jumping. If you think of Austen-Uhlenbeck or any other Brownian motion-based model, they are all thinking about independent changes in each one of the branches. This is also true for the Markov models with or without hidden states, biogeographic model too. So if the difference between bifurcation and budding is the recognition of the ancestor lineage after speciation, then if we assume that these branches are independent, what's the point, right, of recognizing the lineage before and after speciation if the model is going to break that idea, right? Then we really cannot include in these models the idea of bifurcating that way. One way, however, that we can think on these effects is if they can be dependent on the age of the lineage, right? That might be an important factor, make an impact on comparative models from the point of view of being bifurcating or not. So here is an example thinking about competition. I have these 4 species. Here on the middle, we have this progenitor lineage with 3 daughter species. And here on the left, what I have is a plot showing morphospace, so the traits of these lineages, and the other one, geographical distribution. Now, the idea here is that these progenitor lineage is accumulating age over time, so it's getting more mature, right? It's a lineage that didn't get extinct. We expect it to be stable in a fitness landscape, so occupying a single optima, right? And the new daughter species, we would expect them to have smaller population sizes, to be pushed away from the progenitor lineage by competition, and also having the opportunity to switch to a different evolutionary optima, right? Or get extinct, trying the process, or merging back.
which would be failed speciation events, and we wouldn't see them anyway. This same process might also happen in the morphospace. These lineages could be pushed out from the morphospace because they are adapting to different conditions, right? So this idea of competition kind of depends on this idea also of lineage age over time. All right, here's another example that Matheus showed, and here thinking about how this can impact how we estimate ancestry states. So here is an example in which we have these lineages in yellow being inland lineages, and the peripatric speciation created this opportunity for these lineages in black here to change to an evolutionary optima that might be very different. If you think about plants, for example, here in the coastal region, we might have very different soil properties, very different ecological factors that can change them. However, the question here is what happened with this history in which the black trait is the new trait and is evolving multiple times when we think about ancestral state reconstruction, right? If we do a parsimony reconstruction, this is what you're going to get. You're going to get the black state as the ancestor for most of these nodes. If you do like Killyhood, it's interesting here, but keep in mind that this example does have very few lineages. So this affects how certain we are about the ancestral states. But if you think about this uncertainty, is actually closer to what we had as the truth, simulated truth, than parsimony reconstruction. Although parsimony reconstruction will give you more certainty, will say yes, this node is definitely black, right? So I think that when we recognize this progenitor lineage after the speciation as an idea of budding, these long-lived lineages can occupy this stable optima, right? And push the other ones to different optimas, which will increase rate of extinction, and eventually they will occupy different options, which is a process that we have in macroevolution in our partners that we see empirical data too. Now, one way that we investigated this type of question is to see how lineage age-dependent processes can impact our models of trait evolution. So here we're thinking about continuous trait evolution. And here on the right, what we have are different lineages colored if they are progenitor lineages. So this green lineage, for example, here continues after this event of speciation and continues after the second one too. So here we have a budding event, and here we have another budding event. But in the same tree, we also have bifurcating events, which are just not colored. If we follow the same green lineage here on this other side, what we have are how fast they're changing over time. So here on red, we have fast rates of change, but then as they increase in age, we go towards the green colors, so they decrease the change. And here we are trying to emulate this transition from fast evolutionary change after splitting out and then stabilizing towards stasis, really. So when we do that, we explored the effect of budding together with this slowdown, depending on age, and also not budding. And what's going to happen is that if we don't have budding, then every time there's a bifurcation, these two new lineages will evolve fast and start to stabilize after they accumulate age. If there is budding with the recognition of the progenitor lineage, the progenitor lineage will already be in a slow rate of trait evolution over time, and the new lineage will have a fast jump and then accumulate age over time. Now, just to summarize what we found with this, is that first, if you do model adequacy tests from Pennell and colleagues to 2025 on American Naturalists, it's going to show you that short branches accumulate a lot of change, right? And that makes sense when we think that we are putting a lot of change close to nodes. The other thing that we find is that together with many other types of empirical data, OU is the best model, but with very long half-lives. And we have short phylogenetic half-lives when we have strong age-dependent processes, as we would expect. Now, just to finalize here, I think that thinking about budding speciation and lineage age and this recognition of the long-lived progenitor lineage being stable can be very interesting in cases that the distribution of the traits across the tips really challenge what we think about that trait evolution. So here is a case, and imagine that we do have fossil data showing us that the presence of these traits, so the state black, exists in the fossils. Also makes sense that once this trait is lost, it will not be gained. And this can come from external data. Maybe we are working with a trait that the physiology is really complex. Maybe we're working with a genomic structure that gives us evidence that gaining that trait again is hard. But we still see this distribution. So what will happen? If you try to do this ancestral reconstruction, you're not going to get a black as these nodes internal. So maybe the progenitor lesions can carry this trait towards these nodes, even though the ancestral reconstruction will not do that, right? That's what I have for today. Thank you so much for your attention, and that's all.
Okay, thanks Danielle for that really interesting talk. We're a little bit over, but we have our long panel at the end, so hopefully that doesn't make anyone late for anything. Our last speaker is Dr. Sasha Gavryushkina, going to talk about the more theoretical aspects of estimating these budding phylogenetic trees. So please take it away, Sasha.
Hello, everyone, and it's my pleasure to present at our Budding Speciation Symposium. And thank you very much to organizer Bruno and Matheus for making this possible. Today I'm going to talk about the theoretical aspects of inferring budding speciation trees, and mostly about stratigraphic range facilized growth depth model. So the growth depth models and growth depth sampling models, there we have this additional sampling process, and in macroevolution it corresponds to fossil sampling. It is a very common model in phylogenetic. It is used for describing this speciation process and for inferring phylogenetic trees as a prior or as a model of speciation. And it has been used for dating analysis, where we can have we have molecular data of extant species and morphological data of cells and the ages of extinct species. And then we can use all this information and single inference to infer dated phylogenetic tree. And then we looked at this model, the way it was used. So this is a sliced graph depth model to infer in this total evidence approaches to it didn't treat the fossil sampling process accurately, because instead of using all the fossils of the same species, we just use a single fossil occurrence. So for example, these three fossils could belong to the same species, but instead of telling this to the inference, we would just say there was just a single fossil for this extinct species. So we decided to solve this problem. And what is one characteristic of both DEF models, that they don't distinguish when we have those split speciation events, we don't have any assignment of species to the lineages, and those splits are considered symmetric, and we don't know anything about what was happening there. I mean, we don't assume anything. So when we try to introduce to this extra information that we have multiple fossils per species, it appears it became important to actually specify what's going on at the speciation event, which of the lineages after the speciation event belongs to which species. And this is where we introduced we introduced a mixed speciation fossilized growth depth model with stratigraphic ranges, but practically we can only use the budding mode because this is the only model that's implemented. And then in this model, which we call stratigraphic range fossilized growth depth model, we already model species assignment. So if we consider this phylogenetic tree at each speciation event, we see that the left lineage is a continuation of the original species, and here on the right we have a new species. And again, we have fossil sampling, and since we already know which segments of the tree belong to the same species, we can then assign fossils to different species. And those segments between the first and the last occurrence of fossils of the same species are called stratigraphic ranges. And as usual, and we cannot, we usually do not observe all the species. We can, and instead of inferring this complete history, we infer so-called sampled or reconstructed trees in which we don't observe, we don't have this unobserved species. And then it also appeared that we don't have to include all the fossil samples in the inference. First of all, it's impractical. Second of all, we can analytically integrate out those intermediate species, and for the inference, the aim of which is inferring phylogeny, dating it, and inferring the speciation rates. It's enough to only have the first and the last occurrence. This model has been implemented in these two. So it's if you have fossil the ages of the first and the last occurrence of extinct species, we have molecular data of extinct species and morphological data of extinct and extant species. We would use this model for the inference. Kate Schumann, who is a PhD student, and she has given a talk earlier today on the extension of the stratigraphic range FBD model, where we can also allow the speciation rates to change through time in a piecewise constant manner. So we have several intervals, and each interval has this speciation rates fixed. So this model is also now available for the inference. And now I'm going to talk a little bit about the implication of these inferences there. We directly assume budding speciation. And this is one side of such inference, and the other one is that we can correctly assign can correctly include multiple fossil samples per species. So the first thing I would like to mention is identifiability. This is then having different parameters, different birth depth sampling rates, if we have different parameters, those different parameters produce different distribution. That means that theoretically, given enough data, given a tree, we could infer those parameters. And there have been a lot of worries about the identifiability of the Brogdorf model recently, and we have shown that the fossilized Brogdorf model is identifiable. If you look at the stratigraphic range fossilized Brogdorf model, then if we keep all the fossils, the identifiability results would translate to the stratigraphic range fossilized Brogdorf model. However, once we remove the intermediate fossils, and especially in the non-constant traits, I don't have the answer, although the simulation study that we have performed show that in the piecewise constant cases, the rates are identifiable. And here I would like to switch to another important implication of using these two different types of models. So in this case, this is comparing a fossilized Brogdorf model. So one species per sample, no assumption about the types of speciation and stratigraphic range fossilized Brogdorf model. And we can see this is our study, one of our case studies on penguins, there we infer speciation rates using these two types of models. So this orange and brown are just FBD, and these green ones are SRFBD model. And we can see that for the sampling proportion, which reflects our sampling rate, we observe this difference in the inferred rates under these two different models. And although we do know that we don't include all the species in the BD analysis, our sampling rate will be underestimated, and we can correct for this. However, because we do this jointly, we do the analysis jointly, and the rates have influence on each other, they will, once we accurately model the sampling process, because it also influences the inference of the other rates of this speciation and extinction rates and dates and everything else. We can see some changes in these rates as well. And this is important to model the sampling process correctly, because they'll influence other rates and other parameters of the inference. There's another example from this same study where we analyzed canids, and here we have more drastic change in the sampling proportion estimates and also in diversification and turnover rates. So another, so here I will just talk a little bit about these piecewise constant rates. So if we allow rates to differ, and then these are some results from Kate's analysis. There she used a different way of splitting the duration of our clade existence differently in the intervals. There the rates vary. So one way is to equally split. So these blue ones are for equally sized intervals, and then we also use chronostratigraphic way of splitting. There we try to to match geological periods of time, and we can see that when we, first of all, when we use allow rates to vary, we often are outside of the credible. This red one is the constant rate model. And so in many cases, the rates differ from what would be infer if we assume the constant rates. And this was for the penguins, and this is similar, the same type of models for crocodilian analysis. And here, what's interesting is to see if we look at this, if we take the most recent interval, this small interval as a separate interval, we can see how extinction and something are higher at the last interval. So one more thing I will mention is that I have talked about how this different modeling influence the speciation rate inference. It's also influence dates and topological relationships that we infer. And this is just one interesting example from Kate's analysis. There we analyzed extinct and extant crocodilians. And we can see here two clades. This one is Gavialdi family, and then Phylogenies genus over here in purple. And if we don't use any dating and just do phylogenetic analysis, these two clades are placed as sister. However, if we use deep dating, so we also add the ages of the fossils to the analysis, the Phylogenies clade is inferring more, it has this more basal position. And this is confirmed by Kate's analysis when we use chronostratigraphic interval split. However, if we use a different split, one of the species from Phylogenies clade recovered as a sister to all the other Gaviales in this analysis, and their support is at least 62%. I think I am very close to my time. So I'll just briefly explain what we are continuing working on. So first of all, it's great we can use those models. We can more accurately model fossil sampling and model body speciation. However, there are efficiency issues as usual with Bayesian inference. And in this case, I think introducing new operators for this new type of stratigraphic range trees could help. And another thing is that we would like to have a summary tree for our posterior distribution of stratigraphic range trees. And there has been recently some advances in this area. Lars Sperling with co-authors introduced a CCD0 map tree that has desired characteristic of the summary trees. And we would like to also extend this to stratigraphic range trees. And just briefly, what I mean by desired characteristic of the summary tree. So what we want is that our summary tree reflects the monophyletic relationships, or represent the clades that are supported by our posterior distribution. We would also like, because now we have this additional level that we have orientation, we would like it to also represent the orientation. And additionally, we also have ancestry now, like in this tree, species 3 is ancestral to species 4, species 1 is ancestral to these 3 species. And desirably, we would like that our summary tree also represent this relationship. We also wanted to be an efficient algorithm to find such trees, and as with, especially with morphological data, with such inferences, we will have variance. So we will often have very uncertain tree distribution. So there will be many trees in that real posterior distribution, but posterior sample is restricted to by size. So we would also want the summary tree to have the possibility to be outside of the posterior sample, which is CCD0 map tree fits for. This is work in progress. We don't have any results, but this is what we are aiming at. That's all. This is the team. Thank you very much. Sorry I went over time.
Thank you so much, Sasha. Um, yeah, this was a really nice set of talks, and yeah, as everyone knows, we have some extra time, so we were just planning on doing a little panel, letting people ask questions they have for our speakers, and whenever we have lows, I have some questions I've been collecting throughout the talks as well, that I can ask. So I guess maybe a first talk we could ask is, Sweeting, um, Sue posted on the Q&A chat reasonably early in the symposium, and I think it's an interesting discussion question. And you guys the speakers can let me know if you want Sweeting to clarify. But the question was if we believe there is any non-budding speciation without hybridization. They said, be it allopatric or sympatric, bottlenecked or not, all speciation are bifurcating unless hybridization is involved. So I guess what seems is that they are wondering if hybridization is going to be involved in speciation, any speciation that is not bifurcating. Yeah, so if anyone has any thoughts about that, I don't know how we want to structure this. I was just going to let the speakers talk.
I talked a little bit about things, so I can say my take, which is I think that we're learning that hybridization is remarkably common across all organisms, or across all species pairs, and that I don't really see too much of a reason to think it's particularly special in the bifurcating in sorry, in the budding case. Perhaps the biogeography might provide slightly different opportunities. It seems like another thing I've noticed in my studies of plants, but I don't want to get too far, is it seems like I've largely seen unidirectional gene flow from the budded to the sort of mothership, as I call it, in that it seems like maybe the bud might have more intense local adaptation or have some sort of broad sort of thing to keep together. So I don't know if I would say that there seems to be any special I don't think there seems to be any sort of universal special relationship about introgression and budding speciation, but it seems like there could be a whole bunch of cool questions that might arise about how if and how introgression happens differently in buds than in a clean bifurcating model. I don't know if anyone has any deeper thoughts than that.
I mean, like, budding fits very nicely into a peripatrical speciation, right? Um, I agree. Sometimes I think like, okay, like what do I need to observe to reject budding? And I don't know, somehow maybe other people know much more about me. I'm the beginning of my career, but I think it's really hard to think on something that would allow me to reject like budding happen. So maybe budding isn't hard because bifurcation is kind of specific, right? Like um and it's a matter of how close you are from like a perfect bifurcation. And in many cases it may not be like in places like islands and caves, maybe in like some small um river basins too. Some these lineages got captured that they don't die necessarily or they don't die immediately. And we and so that how far is that from budding? And you don't need like any special biology for that to be a small subset of the genomic material of the species that is now evolving and it can integrate or communicate. It might also not persist through a very long time scale. I think a lot about that, and I wonder if the special part is that a lot of these lineages that bud off, they don't survive because they are in relatively small places, or like if you are in a tiny island, you have a higher I mean, like a higher extinction probability by the sheer fact that you have a smaller range. Um, so from that perspective, that's the thing that like separates the mothership. When you think about the traits evolving the tree, like in Caetano said, the mothership has a larger range, so it has it's basically buffered against extinction relatively to the small bud that lives in this tiny place. And if anything happens to that tiny island, the bud disappears, and we will never know that it happened. Does anyone else have any other ideas about how special budding is?
I mean, I think an interesting thing to, I don't know if I'm just supposed to be the moderator, but I'm gonna give my thought. One thing that I've been thinking a lot about budding and bifurcating is that I think, as a statistician, what really interests me is how, what kind of data resolution do we need to separate, we have, regardless of, if God could come down and be like, this speciation was definitely budding versus definitely bifurcating versus whatever, how does our data need to look for us to truly detect budding versus, maybe the timing of the divergence was such that our models might end up looking like bifurcation simply because we have, imperfect data, right? And it's just hard to identify which lineage was the mothership, right? So yeah, I think it's gonna always be a little bit difficult to say how special or rare budding is because the data we have and the power our models are going to have to distinguish between these speciation modes are always going to be limited, right?
And sometimes the way that we construct the models to extract the meaning from the data implicitly assumes budding too, and we have to be aware of that, right? So we partition the information in a way that tells us something about budding.
Yeah, just to jump in this one that Bruno said, Matheus now too. That's something that I keep thinking all the time, and it's more like to the philosophy of making things than to the evidence of the data that Bruno was talking about. It's like Bruno said, like what's the evidence in the data for us to think about budding? But I don't think that question was ever made to define it as bifurcation. I don't think we went through a period in which we're going to say, oh, what is in this data to tell us that bifurcation? Well, thinking about comparative methods, right, I find it very interesting that for continuous data like body size, our ancestral estimate is the mean. It's it's a weighted average by the branch length, but it's a mean, right? So we have this idea that whatever the ancestor was, the two lineages after from the most recent common ancestor, they both diverge from it. I don't think that. Oh, I mean, it's not even thinking. If you go back to the early papers, there's no discussion if it was bifurcating or not. It was just a simple model to make it. So it might be even like inverse of what Bruno was saying. Like, where is the evidence for bifurcation?
Can I speak? Hello? Can you hear me?
Yeah.
Okay, thank you very much. It's a very nice talk. I think that what I mean by bifurcation is a little bit out of the context of these discussions because I thought that bifurcation means one become two. And if it's just one become two, no matter there is a stem and there is a branch bud out from the stem, or it's like a stem and fork into two, they are all a process of one become two. So mathematically or topologically, they are all bifurcation. But it seems that in this discussion, bifurcation means that A becomes C and B. And body means A persists and there's one group of B that comes out from A. Did I get it right? Yes? Okay, thank you very much. Yeah, so I guess the question will become that because from the genetic point of view, even when a budding speciation occurs, the mothership can never persist the same forever. So I guess the question will become how different the mother stays the same, you will call it a budding. Thank you very much.
I always have too much to say, so I'm happy to provide my thoughts. I thought I would have nothing to say, but but somebody else can. So I agree with that. And I was thinking a little bit about that when I was drawing up my little cartoon. I think that maybe we're thinking about the process in which you bud off, but I think that if you just have a bud that buds off and then you have subsequently sort of not necessarily homogenization, but close enough so that you're coalescing in this one group here and it kind of looks the same. This other one, you kind of erase a whole signal of budding. I would say that it might have been a budding event, but that like we would, in retrospect, think about that or we would I don't think we would have a ton of evidence to classify it as such, even if the process was different, which is why, in my view, I had that sort of third little thing but off. And then I think you're kind of frozen in time. And I think once you have those three lineages where there's not a clear split, then one doesn't become two, one becomes three or seven or 12 in a way that's not clean. So I think that maybe some of the confusion might be, or some of the things that you're picking up on is a distinguishing between the process that you might be interested in and the long-term signal and outcome of that process. So that's my view, but these people all know way more than me, so I'd love to hear anyone else's.
Yaniv, you mean that like the like saturation extinction will raise the statistical signal for budding? Is
Well,
this is
without an extinction event, I think if basically you get to the point where that mothership all coalesces more recently with
ah
the split, then you won't
mhm,
then the bud, then you won't see evidence of that event anymore. I mean, I don't know what
mhm.
all coalesces mean because there's a genome full of coalescent events, but roughly, you know, so most of that signal is gone.
Yeah.
Yeah, I think that's really like, I mean, I've thought about this in relation to the fossil record. I guess that's what I work on. But and I've thought a lot about like that cartoon where like the ancestor is paraphyletic and then eventually sorts out and is, the two are, if there's only one budding event, are reciprocally monophyletic. But I mean, and I think it's a good, in some ways, like, I mean, I've thought about it because it's a good, I think it's one of the strongest for me, kind of sort of semantic, but like in a insightful way, arguments against budding being a thing over the long term. But I do think that, and I don't really have a good answer to that, but I do think that like oftentimes when we think about species in really densely sampled fossil clades, like forams or trilobites or things like that. Um, like I mean, some the species concepts are messed can be messed up for geological reasons sometimes. Actually, a lot of the time for other reasons, but kind of setting those things aside, like what we're doing oftentimes is tracing through within like one kind of set of localities in one set of like one formation, like kind of continuous lineages that I think often kind of correspond to kind of historical reproductive communities through time. Like I think it's reasonable to think about tracing things through that way. And I think that, and so like, I mean, I guess I've thought about that. For me, that's kind of the main kind of counter against the sorting out reciprocal monophily thing is that like even if that ancestor changes morphologically or even ecologically, as long as it's kind of doing so together, to me that sort of still meets the definition of like that ancestral species sort of retaining its historical continuity that sort of preserves the sort of fundamental character of what we kind of would refer to as budding.
Oh, I'm so sorry. I have so much to say. I mean, another thing is that, both bifurcating and budding are just metaphors that I think help us understand the evolutionary process. And so I think it's great to open up our eyes to these different potential processes, and they hopefully will give rise to interesting models that can help us learn more and interesting perspectives that can help us learn more about biology.
For sure. Isn't part of it too that like speciation is just inherently messy and like So we have these cartoons, and to me, I think a lot of like the argument against bifurcation that makes the most sense to me is I think like Yaniv, I really liked how you like explicitly laid out the kind of phylogeographic, models there where it's like to me it is like explicitly tied to this vicariance thing. And like if that's the case, like that's a much it becomes to me a more useful kind of cartoon to if you think about like if it's like vicariance versus other less symmetric modes of splitting.
Thanks, everyone. That was a really interesting discussion. I guess I don't want to interrupt. If anyone has other thoughts, they can feel free to unmute and talk about it. Or if anyone else has questions that they would like to discuss, of course, I'm also happy to mention any of the questions in my list. But I wanted to give space to the audience to say anything if they want. Cool. There's an interesting discussion going on in the chat that I wanted to see if Sasha and Danielle would like to talk about it. Sasha, do you want to mention the question you had, and then I guess maybe we can talk about that. I don't know. That seems like an interesting discussion there. If we want to bring it to the conversation.
Yeah, thanks, Bruno. I was just thinking when I listened to Daniel's talk that if so we have this model of mixed speciation where we can have both types of speciation in the same model. But then if a budded speciation happened, this lineage that budded off, it will have high extinction rate. So if we could introduce this to the model, once the budded speciation happened, the lineage's extinction rate is higher, then this could be additional signal for inferring budding speciation. I don't know if it will be, well, we need to test this, but this is just an idea of, then we have all these data and do this phylogenetic analysis with with fossil data and extant species data, potentially these differences in the rate help distinguish between budding and bifurcating events in the inferred phylogeny.
Yeah, I think it totally makes sense for the model, and inference of where budding events could have happened in the past is really the most complicated part on this whole conversation for both diversification rates and trait evolution. I think the idea of budding having higher turnover, I think, like both higher speciation and higher extinction, like I think it makes sense. Now, would that model be different from just heterogeneity in rates of diversification across the tree? I think that would be the tricky part. Would that generate any pattern different from what we already see with some clades diversifying faster than all the ones? It's yeah. It's the hard part to think is can we figure out if we have signal of budding, be it diversification or trait evolution. I think that's the harder part, hardest all.
Yeah, but we can directly allow switches of rates whenever a budding happen. And there's still a question if there is enough signal in this, but could be interesting to test.
Maybe relatedly to this, I could mention a question that I kind of started thinking in the model that Sasha was talking about in her talk of this kind of mix, or the kind of original math that allows for budding bifurcation, and then it also allows for the third speciation type, right, of anagenesis, where like one lineage just becomes another without any branching off. I was wondering, especially for the speakers who talked about the more kind of population level stuff or the more microevolution stuff, because I think it's clear to me at least how anagenesis might have a signal in the fossil record, but I was wondering how we think that that third speciation type could confuse the kind of, you know, if life was easy and we could just distinguish between budding and bifurcating, maybe things would be a little easier. I'm interested just to hear what speakers think about how anagenesis might confuse that signal, right? It might make things harder.
I, Bruno, I have a feeling that even if you simulate, which makes life easy in a sense, right? I think it still would be hard. So, yeah, it's go back to where the signal is.
Yeah, I mean, I think it's interesting to think about because you have if you imagine like the character evolution happening, right? Um, it does seem I mean, obviously, in some cases, it's going to be impossible, right? We are totally dependent, at least on the fossil record side of thing, which is what I'm more used to thinking about. We're totally dependent on, did the mother lineage or the parent lineage leave more samples after speciation, right? Because, a budding event where the parent lineage didn't leave any more samples after, it's, probably indistinguishable from an anagenesis, right? So, but I guess I am, this organizing the symposium and watching these talks is the first time that I've thought about the more kind of microevolution signal of budding on Yaniv's and Matheus's talks. So I guess I was interested in hearing about how do we think that that could mess with, the island stuff or the, more population genetic way to distinguish these speciation modes.
I'm not sure. I have to think harder about. I'm not sure what anagenesis would do, but I'm not sure if it would lead much of a signal except, um, change over time. Like I don't know exactly what we would see from it, but I have to think harder. I haven't thought about anagenesis really at all. So I think finally I cannot talk and somebody else can have their good ideas.
Tomo, in like well-sampled fossil records, do you see signal for that?
For anagenesis?
Yeah, but for the Genesis, it's common enough they would erase any budding.
Yeah, I don't know. I mean, anagenesis is weird. I think it's like in the fossil record, I normally think of it as being kind of like not like more an artifact of, at least in the groups that I've thought about this most with, is kind of an artifact of stratigraphers finding it convenient to put different names in things found in different geologic intervals. And so it's in that sense, like I don't really think of it as, I mean, I don't think it's a mode of speciation. I think it's just a mode of, it's just morphological change. And so, like statistically, I think it matters for the reasons obviously that you said. I mean, I think in well-sampled fossil records, usually, according to my super unscientific survey that I just made up, it's usually pretty low. Like it's a pretty small proportion that you could reasonably attribute to anagenesis if you just like analyze stratigraphic ranges, in most cases. But yeah.
Nice. Yeah, I guess we only have 2 more minutes. I don't know if anyone has any closing thoughts or any kind of final, quick questions or discussion topics.
If we keep working as a group, we should like pitch part of any conceptual paper in the micro to macro bridge. Like it's like budding being part of that because people want to hear about that recently. And I think at least thinking about like the the beginning of the budding speciation has everything to do with this sort of question, right? How species come to be, So just a thought, if we keep it a group.
Yeah, I think that would be great. What Matheus is referring to, if for those who are not part of the group right now, is that the intent of the Society for the Study of Evolution when putting together these groups is that eventually we would, aspire to kind of turn this into a special issue of evolution. So I guess this is with a minute left, a good time to kind of close this off and say, if you are also interested in budding speciation, please feel free to contact us. If you know any of these speakers and it's easier for you to contact them, please do. It's very easy to find all of our emails online. And with that, thank you so much for all of our speakers and their contributions to the symposium, for the great discussion, and for everyone who came to watch it, and to SSE for opening up the space for us. It's been a real kind of whirlwind to organize this group with people in 3 different continents and basically impossible time zones, but I think it was really worth it. Please make sure to come and watch our session tomorrow. We're gonna have 4 more really interesting talks, and including Tomo and myself. And yeah, Matheus, any closing remarks?
See you guys tomorrow at 9 a.m. for more budding speciation.
That one will be early.
Now we sleep early.
Yeah.
Okay. All right. So bye everyone.
Thank you so much, everyone.
Thanks.
Thank you.
Yesterday, and today we are going to have a more paleontological session. I want to thank again the Society for the Study of Evolution for giving this a space and also supporting our activities as a research group more broadly. And with that, I want to pass the microphone to Tomo. Please share slides if you want, Tomo, and you all have a good symposium.
Sweet. Thanks, Matheus. Hey everyone. So yeah, my name is Tomo Parins-Fukuchi. I'm going to talk a little bit about just a couple of projects that I've sort of been pushing over the past a couple of years, dealing with budding patterns in the fossil record, and kind of trying to understand the kind of significance of what happens to morphological variation during those events. So first, just to start, in the fossil record, it's pretty common to find cases where a single ancestral species or lineage gives rise to multiple descendants over kind of a long period where it is extant. And so this is pretty common, especially in well-sampled marine groups like trilobites, as is pictured here. And the way we think about that, I think, is pretty often straightforward, sort of when just thinking about tracing patterns in the fossil record, but then when mapping those to phylogeny and trying to reconstruct the patterns, the resulting patterns of ancestry within a phylogenetic context, that can be a little bit more complicated. But I mean, I would say probably not too much more complicated, but maybe just a little bit. And we do tend to see this a lot, especially we would see this a lot using older approaches like stratophonetics that were sort of common before sort of cladistic approaches and stuff we do now kind of took over the field way back in the old days. But we might ask, using this sort of updated methodological view and kind of framework that we have now, how common this really is. And I think it's, I'm going to kind of only talk about this briefly. I've looked into this a little bit. Some other folks I know are also working on this, and kind of, I think it's, an area of active and interesting development. But one thing that I found is that if we just generate sort of a theoretical expectation for for how often we should expect to see not just budding, but cases where one species gives rise to not just one, but like a couple, two, three, and more descendants that I've sort of self-indulgently called super progenitors, kind of modifying a term that was I first read from a paper by Danielle Caetano, who was spoke in the session last night. But that in general, we should expect, especially in a well-sampled fossil record, like, in these four clades, for example, crinoids, echinoids, forams, or trilobites, other kind of similarly preserved things, that within those clades, probably at least about 10 to 20% of the species we should expect to give rise to at least two sampled descendants. So two descendants that are then also sampled in the stratigraphic record. And so going from that, like if we expect that to happen, what I'm kind of interested in is thinking about what evolutionary processes might be associated with those budding events, and then what patterns might manifest from those processes. And so thinking back to it, one thing that I like to think about is sort of just what we expect to be going on. So pretty commonly, and I think uncontroversially, we expect budding to usually be associated with asymmetric population sizes. So geographically, often it's linked with parapatric speciation, kind of as opposed to vicariance speciation. Yaniv gave kind of broke this down nicely in the session last night. But yeah, so going from that, so if we expect there to be sort of a widespread and larger population size ancestor giving rise to a much smaller to a descendant that sort of breaks off within one sort of isolated region. And as I would say that I don't think it has to be parapatric, but anyway, so one smaller subpopulation, then that we might also expect that asymmetry to give rise to certain patterns in with regard to what happens to the morphological variation that is present within the ancestral lineage. And so to me, that sort of thinking that sort of process through brought to mind this area of research that a few folks have been working on in kind of the molecular phylogenetics and popgen world, probably pushed most notably by Matt Hahn's research group over the past 10 years or so, where we may expect at speciation, or we should expect actually kind of regardless of what the specific mode of speciation is, there to be some division of ancestral variation when speciation happens. And so some folks in Hahn's group have been describing this as sort of a sieve that happens. And so when speciation happens, there's ancestral variation that then becomes filtered or sorted among one or more descendants. And that has downstream consequences in what happens to that descendant post-speciation. And so, yeah, again, I think that's pretty, I think that, we expect that to happen regardless of what the mechanism is or the specific mode, because, speciation kind of necessarily requires some sort of division of the ancestral population. I think that budding is particularly compelling, though, in thinking about this specific pattern because of the kind of potentially extreme asymmetry that we expect to happen during budding should cause this kind of sieving effect to be even more pronounced if a lot of budding is going on. And so to look at this, I was interested in sort of formalizing a phylogenetic model to describe what happens to ancestral variation during speciation. The first place I went, so kind of the first step in this was borrowing a model that already existed from the kind of biogeography world. So specifically, I looked at the DEC model, which actually describes this pattern really nicely, where for those who are not familiar, it models geographic range evolution. So species can be in one or more geographic ranges. And that during at a phylogenetic node, which we model as a speciation event, that ancestral range is subdivided among descendants. And so that happens according to kind of what in the deck parlance we refer to as inheritance scenarios. I just sort of took this model, made a couple of simple modification. I added a couple I actually modified the set of inheritance scenarios to correspond to to better describe this asymmetric speciation mode. So instead of an ancestor splitting into two, an ancestor that gives rise to one, and then the sort of inheritance happens just between that for that one descendant. And then just kind of like recast the sort of application and thinking to deal with morphological evolution. So the evolution of polymorphic morphological character states rather than discrete geographic ranges. And sort of went from there. And so it divides evolution into two components. So we call that cladogenesis and anagenesis. So cladogenesis would just be change that happens during speciation, and anagenesis we just refer to as any of the morphological change that happens within a lineage. And so it's simple. When a budding event happens, this sort of modified model says that the ancestor stays the same. So it just continues and it can evolve anagenetically within the ancestor, but at the speciation event, the descendant immediately inherits some subset of the ancestral variation, or it could be all of the ancestral variation as well. And that anagenetic change is kind of just your standard Markov process that like the MK model and all that kind of also describe if you're familiar with that. But the only difference being that it deals with polymorphic character states as well. So I was interested in applying this to this group that I'm really interested in, that I really like, that's I think of really classic importance to evolutionary biology, actually generally, not just paleontology, called Micraster, which is a Cretaceous genus of echinoids. They're super well preserved. In general, irregular urchins are awesome to work with because they bury themselves. So they kind of fossilize really, really well. And biologically, they're also kind of interesting because their phylogeny has been really sort of not super well characterized for, over a century, despite the fact that it's sort of been intermittently looked at in a series of really important, I would say, studies. And part of the reason for that is because there is just a lot of polymorphism within the lineages that have been named, and sort of further biological interest. A lot of that variation seems to be functionally relevant and sort of has ecological significance, it seems. So I just applied this model, put together like a boneheaded, really simple tree search. And kind of like there's two takeaways that I'm just gonna kind of speed through here, because these conference talks always go by way too fast for me. So one thing is that we do find a pretty extensive pattern of budding across Micraster. I didn't note it when I was describing the model before, but actually bifurcation can also happen under as I kind of as it's implemented here. The set of stratigraphic ranges and the pattern of phylogeny is pretty consistent with this sort of budding pattern. Also of note is that we do see a couple, especially one lineage in particular, so Micraster cordicudinarium here, which it would be, which gives rise to, I think, 5 descendants. And so there's a little bit of asymmetry in there's a lot of budding, but then also that budding shows some asymmetry where there's, in this case, one lineage that gives rise to a bunch of successive lineages. So another takeaway I found was that variation seems to sort of proceed evolve such that older, more variable species tend to give rise to younger, less variable species. And so on the x-axis here, I have the number of character states displayed by each species in the phylogeny. And then the y-axis is the number of character states displayed by its ancestor. And so, on average, we tend to see that the descendants tend to be a little bit less variable than the ancestors. But some of that variation that's lost is being replaced by anagenesis, so by the addition of character states within those descendants. And so I don't have time to get into the biology too much as I'd like to, but there does seem to be a lot of ecological partitioning happening between these species within Microaster. So they sort of each species, and some of them actually occupy multiple region areas, but they sort of are associated with burrowing at a different depth within the sediment. So they sort of carve out separate niches in that way. And so I'm kind of interested in this pattern of ecological speciation that could be happening where new species sort of change their morphology as they radiate into a new niche. And so I was interested in asking this question as to whether there's a bias where descendants tend to inhabit different display less similar or more similar character states from their ancestor than you would expect, which you would expect if either they tend to be radiating out, into new niches or into the same niche, but sort of there's like a bias there. And in short, we don't see that. The things tend to be sorting more or less randomly, which seems like a negative result. I guess it kind of is, but leads me to the end here, which is that where I am interested in exploring this as a mechanism, this random inheritance as a mechanism for possible differential speciation and extinction, or extinction of the incipient species. And with that, I'll take any questions during the panel later. Thanks, everyone.
That was pretty awesome. Thank you, Tomo. Well, I definitely will pull more discussion about this later. But now, Rachel, the microphone is yours. Thank you. So are you ready, Bruno?
Yeah. Thank you
Alright,
so much, Rachel. That was a really interesting talk. And yeah, thank you everyone for coming to. Oh, can I start? Sorry, is it? I guess we gotta.
Yeah, you can.
You think? Okay. Thank you everyone so much for coming to my talk and to our symposium, and to the Society for the Study of Evolution for opening up the space for us, and all of our amazing speakers for their contributions. I'm going to be talking to you today about one of the more theoretical aspects of this budding speciation mumbo jumbo, which is tree metrics. So, what are tree metrics? I don't need to tell you that phylogenetic trees are simply hypotheses of the evolutionary relationship between taxa, right? So, if I show these two trees, we can agree that they represent different relationships about different hypotheses about the evolutionary history between these four taxa. And we can qualitatively discuss what these differences are, but it would be really nice to have a quantitative representation of these differences, right? And that's where tree metrics come in. They're very useful in many theoretical and empirical applications. And there are many options in the field right now, but they all have the same pesky assumption that we have been talking a lot about in this symposium, which is bifurcating speciation. We assume that once the parent lineage generates two other lineages, it goes extinct, and the internal nodes in those trees represent undescribed hypothetical taxa. There's two models in the last 15 years that put these assumptions on their head. Rachel and Sasha in the last section talked a little bit about these models. The fossilized birth death models allow for the inference of trees with sampled ancestors, right? Fossil samples that left sample descendants, meaning our internal nodes now might represent described taxa. And this tree is going to be a distinct biological hypothesis from this one, right? More recently, last year, or the first time it was implemented was last year, the stratigraphic range FBD model allows not only for sampled ancestors, but also for these budding speciation nodes, where the parent lineage is allowed to continue living after it has generated more lineages, right? If you were in our session yesterday, Sasha talked very deeply about the theory behind this model. It also allows for stratigraphic ranges, which are, in the case when you have multiple species, you have more than one occurrence per species. We're not going to talk too much about that, but it's good to keep in mind. Now, it would be interesting to have metrics that account for both of these features, right? Because it would allow us to use all the available information in our trees. SR and SA trees just have inherently more information than traditional bifurcating trees. For example, if you come to my in-person talk in a month in Cleveland, I'm going to talk about my simulation study comparing the SR-FBD and the FBD model. And for a simulation study like that, it would be really interesting to have a metric that takes into account all of these available features so we can get a thorough idea of the performance of these models, right? It is unclear, however, whether it's better to compare topology and these extra features jointly or independently. Do we redefine what a clade means in this context for these metric definitions, or do we consider these two kind of aspects independently? And real quick, what I mean by better metric here, there's a lot of different features that we desire in our tree metrics. The one that I'm kind of focusing on in this case is just what I consider to be the most important one philosophically maybe, which is that you would want the distance between two trees to be roughly proportional to how different those underlying evolutionary hypotheses are, right? So if I show you these two trees, they are fairly similar. Topologically, they're the same, right? Even though we have A as a sampled ancestor here, meaning that they represent technically distinct evolutionary hypotheses, but they're more similar to each other than either of them would be to this one, right? Which is just a slightly different topology. So we would hope that a good metric would place these two trees at a lower distance than either of them would be to this third one, right? That's one aspect of metrics that we were going to explore today. So the goal today is to discuss the extension of available tree metrics to both SA and SR trees and compare these two different possible approaches on how they lead to better metrics for our comparisons. I'm going to be focusing on the Robinson-Folds distance today. RF has a lot of drawbacks, but it is by far the most used metric in the field. And even though we can discuss its pros and cons all day, I think it is a very good metric to do just a simple use case study today, just to illustrate to you these different patterns. So RF is simply defined as the symmetric difference between the sets of splits in two different trees. So for example, if we look at the same trees I've been showing you for a while, we can build their split sets, right? For tree 1, we have the AB and the CD splits, and for tree 2, we have BCD and CD. There are two splits in these sets that are not shared between sets, so we say that this has a distance of 2 out of a possible 4, which is simply all of the splits that are not the root between the two trees. Now, one main drawback, or two main drawbacks for the RF distance is that it is easily saturated. You can get the maximum distance between two fairly similar trees, and it has a pretty low resolution, right? A fairly small set of unique values that we can have between two trees. Still, again, I think it is a useful, straightforward metric to extend so we can illustrate these approaches. So let's talk about extending RF to a tree with sampled ancestors. A first idea, which I'm calling here the joint approach, is to simply redefine what we call a split to include its most recent common ancestor, right? Whether it is a speciation node or a taxon. So if we're building our split set, now we have the most recent common ancestor of a split in its definition, right? Either nothing or one taxon. We can do the same for tree 2, right, with C as its sampled ancestor of the split CD, for example. And we also need to take care to include the root in our split set because it is, we might have a sampled ancestor in the root. If we do this, again, we have four splits here that are not in both sets, so we get a distance of 4 out of a possible 6 because we need to include the root split in this maximum now, right? Of course, this might be obvious to some, but it might make the RF distance even more easily saturated, right? So that's a problem, and that's what led me to also consider a separate idea where we look at sampled ancestor status and topology independently by considering trivial splits, by which I simply mean the single taxon splits. So if we look at our split set, now we just have our two splits without considering our sampled ancestors. We add our single taxon, and we also add a little tag for each taxa that are sampled ancestors, for example, A here. We can do the same for our second tree, keeping in mind that if a taxon is a sampled ancestor in one tree and not the other, we'll consider that as different splits. So in this case, we would have a distance of 6, right, out of a possible 12. We didn't really change the distance that much. If you notice, what we mostly did was change the maximum by including the possibility that the single taxa would change as well. So this might lead to a little bit of a better resolution by increasing how different two trees might be. We can apply the same idea to trees that have oriented nodes. So we could, again, jointly consider the orientation of splits with topology. So, for example, in our tree here, we could have D generate C as a split instead of just calling it CD. We can do that for both of our trees and get a distance of 6 out of 6. The maximum distance, again, illustrating that this is a really easily saturated metric. Of course, then we can apply the same idea of we did to SA trees of doing just things independently, where we have our topological set of splits, we have our oriented set of splits, and we define our distance as the symmetric difference of both summed up. So we would have in this case a distance of 8 out of a possible 10. Again, we mostly increase the maximum so that we can maybe hope to gain some resolution here, right? And I think philosophically this is also really satisfying, because in the first case, we're kind of considering the change in orientation as the same importance as a change in the membership of a split, right, of a monophyletic clade, which I think is a little bit unsatisfying, right? We don't necessarily think that different orientation makes a clade more different than simply having different species, right? I think this is more in line with the philosophy of RF and with our intuitive idea of the underlying evolutionary hypotheses. I'm not going to make you suffer through the math when we expand this to stratigraphic range trees, but it is basically just putting those two ideas together, right? Remembering when we calculate our maxima, of course, that these first occurrences are always going to be sampled ancestors, so we need to take that into account. If we look at the joint approach for these two trees, we get a distance of 6 out of 10, and for the independent approach, we get a distance of 8 out of a possible 26. So basically we have more or less halved our normalized distance here. Before, I've been referring to resolution a lot throughout this talk, and we'll explore that a little more systematically for these kinds of trees. But first I wanted to give you one last example with individual trees so we can call back to that idea of, does this metric really preserve our intuitive biological understanding of the ordering of these trees, right? So we have here trees 1 and 2, the same ones I showed you in the previous slide. And in tree 3, we simply took tree 1 and inverted the orientation of every single speciation node except for the one for C first. Now, because these two trees are topologically the same and simply differ in the orientation of these speciation nodes, we would hope that they have a lower distance than either of them would have to our second tree. So let's check whether that's the case. We already calculated the joint and independent distances between trees 1 and 2. For trees 1 and 3, we get 0.4, and that's actually the same value we get between 3 and 2, which is, again, a little undesirable, right? These two, we agree, represent more similar evolutionary hypotheses, so it's a bit of a bummer that we get the same distance, right? Fortunately, when we consider the independent approach instead, we get a lower distance between tree 1 and 3 than 2 and 3. Again, kind of harking back to this idea that maybe considering orientation independently can lead to an ordering of trees, right? A structuring of tree space that better follows our intuitive biological interpretation. Now, this is great, right? Let's talk a little bit more about resolution. To get a more systematic idea of how these different approaches change in resolution, I applied all these metrics to a set of 5005 budding phylogenies from SRFBD analysis on canids. Again, come to my in-person talk at Evolution if you want more details. There were around 12 million tree pairs, and just as a side note, all the distance values here are normalized. For the normal RF metric, just topology, we get a resolution of around 100, again, showing that this distance just has a really low resolution. When we look at the joint approach, considering just essays or essays and orientation, we get around the same resolution, which was surprising to me. I expected that to go down a bit. But I think the real thing to compare here is when we look at the independent approach, especially when looking at orientation, we get more than a twofold increase in the resolution, right, in the number of unique values that our distance can take. It's still a really low resolution, right, for 12 million tree pairs. Obviously, there are much better metrics in the field that we need to explore, but it is really interesting that looking at orientation independently can lead to such an increase in resolution. And I hope I have convinced you that, even though it might be more straightforward to redefine what a clade means, maybe considering things independently would make more sense moving forward. In terms of next steps, I would like to extend some better metrics, right? My collaborator Martin Smith published on these really interesting information theoretic metrics where we are looking at the shared information between splits rather than just whether they are the same or different. We would like to build more systematic comparisons. These are all very preliminary results, just meant as a proof of concept for the symposium. And of course, with the final goal of applying all these ideas to my simulation and empirical studies, and hopefully I've interested some of you enough that you might want to apply these to your data as well. And with that, I'd like to thank you for watching, thank my co-authors and my lab, and again, the SSE and my co-organizer and very good friend Matheus for helping us all organize this very nice symposium with our speakers who had all really amazing contributions. So, thank you.
Okay, so I actually want to like kickstart the discussion here. So it's very interesting that we talk a lot about sampling, right? Like it looks like it's a matter of like observing the data that allow us to tell that something looks more like budding than anything else. And we talked a little about this yesterday when we discussed, okay, like where is the evidence for like budding, but maybe we should, and we thought yesterday that maybe we should flip the question and say, where is the evidence for bifurcating speciation, right? And maybe we want to start with Rachel, but also I want to quickly like go through Tomo and Denise to like which type of data would allow you to say this was a budding event and this was a bifurcation event. So Rachel, do you want to start?
Oh gosh, throwing in there. I think it's really difficult, and I think so in terms of data, like in the absence of any models, like there are some extremely high resolution fossil records, like foraminifera, where the sampling is so good and continuous that people who are experts in the taxonomy really can demonstrate that very likely you have budding, bifurcation, or anagenesis. But in almost all of their cases, it's extremely difficult because of the incomplete sampling. So we need like an inference framework to be able to do that. And so like some of the people that Denise just mentioned, like Sasha and Ugne and Joel Burrito Santani, they're trying to work on implementing these like models that can explicitly account for bifurcation and budding simultaneously. But that'll be the best way, really, when we get that up and running and we could apply it to a bunch of different data sets and maybe try and test it that way. The problem is a big part of that research is going to be whether the parameters are identifiable. Yeah, so that's what I would say.
Tomo, do we want to go next?
Sure. I mean, I agree with everything Rachel said. I mean, I think that yeah, I mean, I think, yeah, the really well sampled fossil records, can offer a lot of insight into this. There's a really nice paper from 2017, I believe, in paleobiology that Dave Bapst published with Melanie Hopkins, where they look at this in a really well sampled trilobite clade. And I think that sort of approach of like basically what they did, which I think, is what I think I would say all of us are kind of trying to do, kind of similar things, building on that, has been to apply a birth death sampling model. And, like it's kind of like the idea would be to see, like, for example, to distinguish between anagenesis and budding, like their kind of approach was to see like how much you'd expect. So if two closely related taxa do not overlap in their stratigraphic ranges, and you apply a model of incomplete sampling, um, does a model where anagenesis happens and the two lineages are not overlapping in their ranges, like in reality, better explain the stratigraphy, or does a model where they are overlapping but we didn't observe the overlap better explain it? And so I don't know. I mean, I think things like that, also delving in really closely and doing I mean, I think a lot of, I don't know if I'm not obviously a geologist, but I also do kind of suspect if you just asked a lot of biostratigraphers what they think, I feel like a lot of them and a lot of their groups would just say, like, as we trace, these, lineages through the rocks, they overlap so much that if I had to guess, I would say it's budding. I mean, I don't think that's universal. And I think Rachel, you had a nice example of a well-sampled marine group that probably does not look like this, but I do think that anyway, yeah, I think stratigraphy is, you know, can provide a lot of evidence here.
Denise, what type of evidence would you see in viruses that would say, okay, this was a bifurcation actually?
I was just thinking that maybe I should have given the counter example as well. One situation that I can think of where it wouldn't be budding is, for example, in Ebola, right? Where it happens sadly quite often that, the patient dies and then still transmits to many different people and then I guess you still don't have a bifurcation because you'd rather have polytomy, but that makes it even more complicated. But I think in most cases you would rather include that in your modeling approach rather than trying to infer it, because in most cases you would have a good idea of, depending on which pathogen you're looking at, how it is transmitted and what's the most common pathway. I want to say that identifiability is definitely a big issue, like Rachel said, and even for viruses where we have faster evolution, I would be careful to claim that we could easily estimate that if you wanted to.
Jump in.
Yeah, I think it's gonna be really interesting. One of the main things we've talked a lot about models that how most models only look at bifurcating speciation, and how now we have models that look at only budding speciation. And I think that it'll be really interesting to ask the question of when can we, like, we'll have more information about this whole discussion of when can we estimate budding when we have an implementation of some sort of statistical model that can estimate both, right? As Rachel talked about in her talk, the full kind of stratigraphic range model that we keep talking about has the capability for that, but all the current limited implementation is only budding. I think it would be really interesting to do like a simulation study of some sort to see how, what's the limiting factor, right? Is it like stratigraphic, like density, right? Do we just, as Tomo said, like, do we just need a lot of like closely related overlapping stratigraphic ranges? How do we, what kinds of data limitations do we need to be comfortable saying like this is a budding speciation or this is a bifurcating speciation? And I think there's a lot of kind of situations where it'll be unclear if we have a true kind of, if we inferred a true bifurcating speciation versus we just don't have the data to infer the orientation, or if we infer the true anagenesis versus we just don't have the data, right? So yeah, I think there's a lot of cool stuff to explore here, and I'm I'm biased, but I think that as with a lot of other questions in the field, it just kind of goes through, you know, the exploration of these really powerful statistical models that we keep iterating on.
I agree. Well, does anyone in from the audience has a question or a comment or wants to jump in? All right. So I want to actually keep going through to this path because I wonder like how much of this is a matter of like rate. So rate of anagenesis versus rate of trait evolution. Can we separate these two things? And versus rate of the bifurcating effects and how we are kind of like changing the denominator, but because usually these rates are per lineage, right? Through time. So how much of that, and maybe it's my ignorance, but like for instance, Richard, how much of that was like just because we're changing the denominator, how many total lineages we're counting? That was a little unclear to me. Maybe it was because I was making my notes here. But and how much of that is like so basically I'm trying to say is like when you say like this was a bifurcating effect, you are changing the denominator of rate of lineages and you are changing the overall rates, right? And how much of that is from like a specific data that is outside from the inference of budding or even maybe the diversification itself that allow us to polarize that event and say, okay, this was actually a budding. Maybe this connects a little with, for instance, a little details of the ecological and functional traits, Tomo, that you talked about in your talk. So I don't know, do you guys want to ping pong a little on this? And maybe Bruno, you can also, because we're talking about how we polarize this and how we like, kind of like shed light on the importance of body to infer the phylogeny in the first place. Did that make any sense?
It made I mean, it made sense, but maybe you could clarify the question a bit.
I'm just like want to like ping pong a little between this idea that like, okay, how much of this is anagenesis, how much of this is rate of evolution, how much of that is like something qualitatively different that allow us to say, no, this is different from just rate of evolution or anagenesis, this is a bifurcation. And how these three things connect for we to like use this to like, for instance, give more resolution to tree space and walk more easily, which connects with the idea of identifying budding as an output of our phylogenetic inference methods.
So you're saying like how whether the speciation mode might have some effect in the trait evolution, and like it might be kind of a confounding variable kind of sense where
Yeah.
when we're just looking at the morphological characters or the molecular data if you have access to that and all that, how can we kind of ensure that we are being able to parse out speciation mode from just other kind of confounding stuff that might lead to weird character evolution inference like that?
Exactly. Like my skepticism comes a little from like, okay, how much of that is just like a lot of trait evolution, but someone will come and say, actually, this is a bifurcating event. But by doing that and attributed the meaning of bifurcation to these events, that for someone else would be just trait evolution, you are changing the denominator and thus you're changing the rates, right?
Hi.
Yeah, I was gonna say.
Can I jump in a little bit here? So I've been thinking about this in terms of if the trait evolution itself is affected by the node, is affected by the speciation event. So if you're thinking about a trait in which you are considering cladogenetic effects, then the cladogenetic effect itself could be affected by budding. And then you could think bifurcation having a cladogenetic effect and budding have a different cladogenetic effect. And the other thing too is that thinking from the comparative methods point of view, that we have done like not a great job including variation in trait values and using too much just species means. If it is a homogeneous process, right, and we are just having diffusion, it might not matter because that node will have that particular value and then it will start an independent diffusion process if it is budding or not, because it's just homogeneous. It would matter if you have some sort of cladogenetic effect. There are some papers that are really interesting that they start putting the effect of population there, trying to go to either individual based modeling or at least putting the idea of the population. So just like variance, which is a simple thing, right? But if you check a lot of comparative methods work, not I would dare say that the minority include just variance, right? Measurement error or standard variation coming from somewhere. That will help us to understand if budding would matter or not. My hypothesis here, and I haven't tested that, is that budding, when we think about peripatric speciation and isolation, right, and having a smaller population size that could be more suffering the effects of drift, for example, variation itself could be inherited differently if it is bifurcation, like clear-cut bifurcation, or budding, and that could create this snowball effect and change how the traits change over time in the budding because of having like a larger variance, for example.
Tomo, do you want to say a little about like the sorting of polymorphism in a mothership lineage, as we called yesterday in the other panel, into its like incipient or daughter lineages?
Sure. I mean, I think that. Oh, I was gonna say one thing actually before I say that, which is that Robert also has a nice comment in the chat here that I totally agree with. And I mean, and one thing I was going to add also, I think in relation that I think relates also to your original question, Matheus, is that I think that so obviously I think stratigraphy is important, but I also think that I also think that in I mean as Rachel you were talking about too, I mean I think geography also offers a pretty useful frame of reference for looking at that. And I think that's actually like when people like Yaniv when he was talking about, you know, in his session yesterday about in these you know, putatively, you know, budding mimulus, you know, species, like geography is a major, I think, component in a lot of those stories at that scale as well. But then, yeah, and I think also, so if you look at by geographic patterns, um, like you would expect to see this asymmetry that's associated with peripatric speciation. Um, I hesitate a little bit to like build a house of cards on that because when I look at my patterns, you know, that the patterns that I see in the clades that I've been looking at, like that deal with you know, this kind of like sorting at speciation events that happens, the subdivision of ancestral variation into descendants. Like I think it's really tempting to want to say like, oh yeah, this must all be peripatric speciation that's leading to this like bottlenecking effect in all these like small, you know, descendant lineages, some of which maybe are probably ephemeral, like, you know, and you can kind of construct this worldview that starts to feel very similar to, you know, the punctuated equilibrium, you know, kind of worldview, you know, that sort of explicitly was built upon, you know, Meyer's peripatric kind of deal. And like my hesitation is that my understanding is that there's not a ton of evidence that that's widespread. And so I think that, and the other thing I've also like sort of, I'm not aware of a ton of published studies that have looked at this, but I've talked to multiple people who sort of like, you know, have sort of tried to basically like done everything they can to try to get things to bottleneck, like put them in a weird island, like all isolate them, all that, and they won't bottleneck. Uh, and so like that confuses me because I think that we do tend to see this subdivision of ancestral variation in the fossil record, but I'm still trying to figure this out. But I'll stop talking for a moment and let someone else jump in.
Richard, please do.
Yeah, no, just as Tomo was talking about parapatric speciation, I was like looking it up and trying to visualize it. Something that we've talked about, some of us on the panel have talked about before is like, or at least I think about this in the context of this topic, is how we conceptualize speciation at like the different levels different. So like I like spent a lot of time using programs like Beast and software, and so I've really kind of my brain has like made my like I've kind of sometimes I think I've melted my understanding of evolution down to these like very simple models. But like, is it possible that different things are happening at different levels, right? Like, so when I think of molecular evolution, like surely what's going on molecular level is always bifurcation. Like I have a really hard time understanding molecular evolution in like a different like like what is budding speciation at a molecular level, you know what I mean? And so it's kind of it's like we've got these distinct processes going on like on the morphological level where I can totally I can totally imagine how budding could happen in terms of like morphology, but like at a molecular level, surely bifurcation is always happening. And yeah, I'm kind of curious if any of the I'm curious what the other panel members think about, you know, how we could take this in a kind of modeling framework. Like we obviously need to do more work to reconcile like the clock model, morphological model, with the tree model, especially these new range models. But yeah, I'm really curious what other people think about that. Maybe, and including Denise as well, because yeah, from the viral perspective, it's also different.
Yeah, I was just thinking for viruses, you do actually get that. And that's what I was trying to explain that, you would have the current host continuing to, have the virus evolve in them. And then you have this kind of budding off into with a new transmission. And there it really depends on the particular viruses, but you often have bottleneck events. And this is why on a really short time scale, it may not matter a lot. But then if you actually have a longer period, then it builds up a lot of variation in the host. And that can look quite different from the variation you saw on the previous host. And that's all on the molecular level, really. Does that make sense?
We did talk about this a little bit with Yaniv yesterday also, where, I mean, you do expect to see this thing, right? I mean, unless the ancestor goes extinct very quickly, at some point it will probably sort out so that at the coalescent level, it is monophyletic. And also, like, you probably expect there to be some maybe significant change in the ancestor, like in terms of its morphology. I mean, so it could just be coalescent, but I think the change could also be ancestral, or sorry, morphological in the ancestor. But then I think one question I have, or one thing I've wondered is if it's like there's sort of you're sort of almost forced to take a like philosophical stance, I think, because then it's like so if you've got an ancestor, the descendant buds off of it, the descendant is clearly its own thing, but the ancestor has remained intact. It doesn't care about the bud that just happened. And say, you fast forward, a million years and the ancestor looks totally different, or even just as sort of coalescently different. Um, But then I think the philosophical question is that ancestry a different species just because it looks different if it sort of maintained its genetic, sort of continuity and cohesion through time and evolved as one unit. I mean, like that we'd consider that probably anagenesis. Um, like I think my stance is that anagenesis is not a speciation mechanism actually, and so the ancestor is still the same species. But I also think that that's like, I don't know, I don't want to like impose that, super, or, like, I guess overstate that because I think it's just so dependent. It's too dependent on sort of like what your species concept is, and then you will go down this whole path of like being miserable, arguing about something you can't test.
I love arguing about species concepts. It doesn't make me miserable.
Me neither, actually.
But yeah, I mean, an important discussion in this case, right? Especially if we're talking about like the differences between estimating these things for, the fossil record versus more recent divergences, right? Like Yaniv yesterday was talking about, yeah, the kind of population genetics approach of looking at these two different populations and saying, maybe this is a case of budding speciation, and that's going to inherently look different from how we would detect that in the fossil record, right? Where we only have access to morphological characteristics, and like it's yeah, it's difficult. Obviously, we've struggled with a long time for like to find a unified kind of concept for species that somehow gets past that, and yeah, I guess it's just something that we need to keep in mind when we're looking at these speciation modes discussions. We're not only talking about species concept but also speciation concepts, and this is all going to interact in really messy ways, I think. So
And sorry, just to also throw another thought in there that's relevant, like so, especially like talking to Shu Dai, who's the aminoid specialist that I'm working with, he's also reminded me that like actually most of the time in macroevolution, we're not actually able to even work with species. Like we're like for inferring macroevolutionary trends from fossil occurrence data, we're usually working at the level of genera. And then there's another interesting question to be asked about like how do these models apply to genera? I mean, we don't even know like how do you simulate a genera? I mean, I can imagine ways of doing it, but like how accurate is that as well? So there's all these like nested concerns.
We just have 4 minutes left of discussion. Does anyone from the audience wants to jump in and bring a question or a comment? Um, I think like thinking about the levels as you mentioned, which is really really important too. And just as a matter of fact, like I got interested in budding because my master's was about age and how age affects extinction risk. And what I found is that actually the shape of the distribution is very different if you look at the same fossils at the species level or the genus level. So I think this does matter. And it will, if it's very important to think about these scales. And then I did a PhD about populations and again things are different, in not exactly the same way, but like definitely I think that like the final level and then trying to like hold that level constant and then trying to sort out things is possibly the easiest way to not go crazy. And even that is hard. Um, but okay, maybe I want to like make a bar open question like what would we lose by completely forgetting about the idea of budding speciation? If what like because like first I started this whole symposium yesterday saying okay like maybe we have to care about budding, but now maybe as a closing question, do we lose anything by never thinking about budding again?
I mean, I think you lose a lot of really interesting information, right? Like these these methods that allow us to actually estimate budding phylogenies, I think you just have more informing, like trees with more data on them for you to get your hands on, right? For example, if you're interested in, I'm really interested in models that test diversification, like trait-dependent diversification rates. And a budding tree would sort of allow you to, not only now you have your observed traits for your tips, but you can also kind of assume the true trait of some given internal nodes, right? And of course that might, be a drawback in some ways because you're adding kind of more assumptions to your model, but assuming that you can get more information on those trees, I think is always going to lead to more interesting kind of questions that you can ask, right? We don't have a lot of time, so I wanted to let other people talk.
I mean, I would shortly just say, like, absolutely, like, we should be talking about these things. Like, we know from work that, like, all of us basically have done research on the panel, like, that shows like these really seemingly minor differences make a huge difference to the conclusions we draw. Like, I mean, look at applying like Ugane's work applying like the stratigraphic range model versus the OG FBD model, like it makes a huge difference. So I think we have a lot to gain by considering the details of all these models and what we really mean about like a sample. And we haven't even talked about like how do we incorporate abundance data. So there's like so much work left to do in the details, I would say.
With Matheus' question, I think that we lose a lot of models that take geographic distributions explicitly. Biogeographical models, even if they don't explicitly say budding, they are using concepts of budding, right? So biogeographical models not always say that first the lineage will become widespread occurrence in both areas and then split, right? We have different events which allow one of the lineage to continue on an area and the other one being a different area. That if you read that way, it is an example of budding. So I think we would lose that. Biogeographically explicit models would not be there.
Any more closing remarks? If not, I think I'll call it a symposium. Thank you very much, all the speakers. Thank you. Thanks for everyone that showed up. They stayed. The whole discussion was amazing. This will remain recorded and will be available on YouTube soon. I will let the people in the working group work. And again, if you are interested in budding speciation and want to get deeper into this, we are an open research group. So show us an email and you can join us and keep talking about budding if you want. All right. So with that, I end the symposium here and thank you everyone again. Thank you the society for like supporting our activities, and I will see you guys in the in-person conference and around. Okay.
Thank you so much.
Bye everyone.